Hello, future PTAs. I hope you guys are doing well. Today we're going to go over a lecture on special sensory systems. So we'll talk about the visual system, auditory, vestibular, smell, and taste. We'll start out with just some fun facts. The vision system, we have a few fun facts about. Vision is our most prominent sense. 70% of the millions of sensory receptors of the body are all visual receptors. Visual receptors convert light into action potentials, and then those action potentials travel from the eyes to the brain where we perceive and interpret the world. It is pretty amazing how 70% of the sensory receptors for our entire body are for these little tiny eyes. We'll go over the eye anatomy. So the eyeball sits in an orbit. It's surrounded by fat, and it's protected by the eyelid. The sclera is the thick white part. It is connective tissue that surrounds the eye. The cornea is kind of the clear part that's on the outside of the eye that's sensitive to touch. That's where you would put your contact in if you wear contacts. The iris is the colored portion. It's behind the cornea. The iris changes the pupil diameter to change the amount of light being let into the eye. The pupil is, of course, the black circle in the middle of the eye. It opens to let light into the eye. It constricts to decrease the amount of light that is let in like a bright conditions, and then it relaxes to get larger and let more light in darker conditions. We also have the lacrimal glands. Those are your tear glands that they keep the eye moist and help wash dirt away from the eye. And then we have 6 extraocular muscles that are attached to the eye that help move the eye, and those are all innervated by cranial nerves. The retina has the visual receptors from the eye. They're all on the retina. The retina is located in the very back of the eyeball, so we're not actually looking at it when we're looking at somebody's eyes. That's what the doctor looks at when they look in your eye. They're looking at the back of the eyeball, the retina. The lens focuses the light so that it strikes the retina precisely. If the eyeball isn't perfectly spherical, then light can't hit the retina accurately and vision becomes blurry. That's what we call astigmatism. So this picture up here shows when you have astigmatism that the lights seem more blurry. They typically have that glare when you're driving, especially at night. Cataracts is when the lens becomes cloudy with age, and the lens can actually be replaced. Seems so scary to me to have that done, but a lot of people have it done and can see so much better. Accommodation is the ability to adjust the lens shape to focus on objects that are closer or further away. So you adjust the shape when you're looking at a book versus reading a street sign. And if you are not able to adjust the shape well, then you may have nearsightedness or farsightedness, just depending on your vision. Photoreceptors, these cells convert light into action potentials. So remember, all of our neurons carry action potentials. They don't carry the actual information, but action potentials that helps convert and relay that information to the brain. Rods are rod-shaped. That's where they get their name. They work in dim light. They're responsible for our black and white vision, and they're dispersed throughout our entire retina. Cones are cone-shaped. That's how they get their names. They give you that clear, sharp visual perception. They work better in bright lighting, and they give you your colored vision. They're more concentrated in the middle of the retina, near the macula, rather than across the entire retina. There are 3 types of cones that are responsible for blue light, green light, and red light. I think that's so interesting because I would have thought yellow versus green. So it's interesting that that's the case. People who are colorblind are actually missing 1 or more types of cones, and that's why they can't see that color as well. Just a quick fact, there are around 7 million cones in our eyes that help with that colored vision, and there are 125 million rods, so a whole lot more rods than cones. The photoreceptors also contain special pigments that absorb light, and these trigger action potentials. Our optic nerve, this is cranial nerve 2, and it connects to each eyeball and contains around 1 million neurons. So there's a lot of neurons dedicated to the eye. The optic chasm, which is that X that you're seeing in that top picture, it's created when two optic nerves cross on the undersurface of the brain, and then information from both of your left visual fields cross over to the right side of the brain, and vision or information from the right visual fields cross over to go to the left side of the brain. Um, the optic chasm relays information to the hypothalamus, the midbrain, and the primary visual cortex. So the axons that go to the hypothalamus are used to establish our circadian rhythms, the sleep and wake cycles that we go through. Those that project to the midbrain form connections with cranial nerves that control your eye movement. So it allows the eye muscles to coordinate with visual stimuli and to track. So if something's moving across, then your eyes can track it because of those connections there in the midbrain. These axons also cause visual reflexes, such as when a flashlight shines in one side of the eye, both pupils constrict. And that is also the connection in the midbrain. And then the axons project into the primary visual cortex allow for us to have that conscious perception of visual stimuli, and they go on to the occipital lobe. Visual perception occurs first in the primary visual cortex, and then it travels to one of the following areas. The visual association cortex allows recognition of objects. The parietal lobe allows location of objects in space. So they call the parietal lobe the where pathway, and the temporal lobe the what pathway, because the temporal lobe has inferotemporal cortex allows to determine size, color, shape, texture of what we see. So the temporal lobe helps us determine what we're looking at. Parietal lobe helps us determine where is it? Is it close to us or far away? Um, Visual perception, of course, gives meaning to what we see. Parts of the inferotemporal cortex are devoted just to facial recognition. We've talked about that before. If those parts are damaged, then a person could have what they call prosopagnosia, where they're unable to recognize people by their face. So they might be able to recognize objects and recognize that it is a person, or that they have brown hair, or what color shirt they're wearing, but can't remember who they are based on their face, because there's a specific part of our brain dedicated to that. Hemianopsia, this is damage to one optic tract. It could lead to loss of vision in the opposite visual field. So this is a common thing with stroke. This picture would show you what this view would look like with normal vision, and then with hemianopsia. So one visual field is basically cut or lost. Cortical blindness is typically caused by damage to the primary visual cortex, where although the information is coming into the eye, there's no damage to the eye itself. The brain is not able to interpret that information, and so um, you're not able to perceive anything that you see. So it's not useful information. Even though you're seeing things, you're not able to use that information to determine anything about what you're seeing. The auditory system allows us to perceive sound, locate it in space, and understand its meaning. It's important for language, and it has many connections to the language centers in the brain. Our ear anatomy, we have the external, middle, and inner ear. So the external just collects sound and funnels it into the middle ear. The eardrum is the kind of separation between the external and the middle ear. It transmits those vibrations from sound waves. The middle ear is in the temporal bone. It contains the hammer, the anvil, and stirrup, or uh, malleus, stapes, incus. They're the tiniest bones in the whole body, and they help transmit the vibrations to the oval window, which separates the middle and inner ear. The inner ear, it contains the cochlea and the vestibular apparatus. The cochlea is the spiral-shaped space within the temporal bone, lined with basilar membrane and contains endolymph fluid. The organ of Corti, it is a receptor and has receptor cells that are responsible for hearing. It has around 18,000 auditory receptor hair cells. The long projections are what we call hair cells. The true name is stereocilia, and it gives them that hairy look. Movement of the oval window moves the fluid within the inner ear and it bends the hair cells. The hair cell movement releases neurotransmitters that activate the vestibulocochlear nerve. So if they're moved one way, then neurotransmitter is stimulated to be released. And if they're moved in the opposite direction, the neurotransmitter is inhibited from being released. And those are what cause the vestibular nerve to be activated and those waves to be transmitted and then perceived as sound. The vestibulocochlear nerve, this is cranial nerve 8. There are hair cells in the inner ear that form synapses with neurons in the cochlear portion of the vestibulocochlear nerve. These neurons project to both sides of the brain. They travel to the brainstem, the thalamus, and the primary auditory cortex. Auditory perception or sound perception takes place in that primary auditory cortex, and then sounds are interpreted in the auditory association area. So the primary auditory cortex is in the temporal lobe, and remember, sound from both ears goes to both temporal lobes to help you locate the sound. Each primary auditory cortex receives input from both ears, that's what I just said, sends signals to both sides of the brain, which is helpful in locating sounds. Many connections between the auditory cortex and the language areas of the brain exist, especially the regions that help us understand spoken language, and we know that as Wernicke's region or Wernicke's area. The vestibular system, it detects the position and movements of the head. It's located in the inner ear. It's one of our 3 balance systems. So the 3 balance systems are the vestibular system, the eyes, and then the proprioceptive feedback from the muscles in our legs. All 3 work together and send input to the cerebellum about where your body is in space. The vestibular apparatus, it has the otolith organs, which are the utricle and the saccule. They are pictured in the semicircular canals there in the cochlea. The semicircular canals detect rotational movement. The otolith membrane has otoconia, which are like little crystals, and endolymph, which is the fluid, gel-like fluid that move when your head moves. So as they move, they bend those hair cells. And when they bend them one way, again, they cause neurotransmitters to be released. So one direction, they activate those neurotransmitters, and then in another direction, they inhibit the release of those neurotransmitters, and those allow the information to be sent to the vestibular nerve and determine where your head is in space. So like the cochlea, the vestibular apparatus contains those hair cells, those endolymph. The gelatinous membrane here is called the otolith membrane. It's covered with those crystals, carbonate crystals, which are called otolith or otoconia. That's really important because those crystals are what can get displaced. They can tear off and move to the wrong area. If they move to one of the semicircular canals, every time your head moves, then those hair cells get really pushed by those little crystals in the membrane or in the canals, and it makes your brain interpret that head movement as like spinning or rotational movement or like a fall. Basically, you're getting the same amount of input as you would maybe when you're riding a roller coaster, but with normal head movements. So those head movements will cause people to get dizzy, really disoriented, may cause them to throw up. And we can, as physical therapists and PTAs, we can reposition those otoconia, those crystals, to fall back where they're supposed to be in that endolymph fluid, that gel-like substance, that gelatinous membrane. and take all those symptoms away, which is a beautiful thing when you can reposition somebody, do a few head movements, and then their symptoms go away, especially the type of symptoms that make them, physically ill. The utricle detects horizontal movements, like taking off in an airplane, and then the saccule detects vertical movements like jumping. So all of these work together to tell your brain where you are in space. More about the semicircular canals. There are 3 pairs. They're responsible for detecting different directions of head movement. So some go this way, and then that way. So the 3 different, they're anterior, posterior, and horizontal are the names of the 3. They contain hair cells and that gelatinous membrane. The membrane is called the cupula. Information from the otolith organs and the semicircular canals all travel to the vestibular nuclei in the brain stem. It then projects to the motor cortex, the cerebellum, the spinal cord to coordinate, balance, and to guide our movement. So movement in one direction, again, depolarizes the hair cells to release the neurotransmitters, and in the opposite direction, it hyperpolarizes hair cells to inhibit the release of those neurotransmitters. And this is how the nerve impulses are generated and travel through the vestibular nerve. Proprioceptors, this is sensory receptors that are in the muscles, tendons, and joints throughout the body, but specifically the ones in the leg help us the most with our balance. So again, those 3 balance centers are the vestibular center, the inner ear, the eyes, the vision, and then the proprioceptors. The input from all 3 goes to the cerebellum for balance and to help with coordination of movements. The muscle spindles help detect stretch. The Golgi tendon organs detect muscle tension. Those, along with joint receptors, are all different types of proprioceptors. They give you a lot of feedback about where your body is in space. The vestibular nuclei is in the brainstem. It descends into the spinal cord and helps control motor neurons. Vestibulospinal tracts are the primary control system for the muscles for your balance. The primary muscles for balance are typically the muscles in the hip, the knee extensors, hip extensors, the ankle plantar flexors, and the ankle dorsiflexors. And then one last comment there, the joint receptors that detect position and motion of joints are primarily in the capsules and ligaments, because the cartilage that makes up those areas can be displaced. It can kind of be squished. It's kind of pliable. And so there are a lot of proprioceptors in things like the meniscus or in the joint capsule that surrounds a joint, because as it gets squished, as it gets displaced in different areas, that gives your body a lot of feedback about where you are in space. All right, smell. The olfactory receptors are in the top of the nasal cavity. As air passes these receptors, molecules are dissolved into mucus to bind to olfactory receptors and create action potentials. Then neurons in the olfactory bulb send messages through the olfactory tract to the limbic system and the primary olfactory cortex. The limbic system is responsible for that emotional response to odors. The primary olfactory cortex is responsible for that conscious awareness of odors. If the olfactory receptors in the nose are blocked by mucus, it can temporarily block the sense of smell. That's why if you have a stopped up nose or a runny nose, you can't smell very well. Smell and taste are really important to help us avoid harmful substances and to provide rich, enjoyable life experiences. So when those are blocked, then you don't enjoy food nearly as much, and just don't have some of that feedback about uh, the about what's happening in your environment. Anosmia is the loss of the sense of smell. And then the sense of smell can return as olfactory cells can regenerate. They're one of the few neurons in the body that can regenerate. And so sometimes people can lose the sense of smell and regain it. You may have experienced losing the sense of smell when you have a cold or something. That's more likely due to the mucus in your nose rather than damage to those neurons. But if you had something like COVID, it may actually cause damage to those, which is why it takes longer for that sense of smell to return, and some people never get it back. Um, Head trauma can also cause a loss of the sense of smell, especially if the ethmoid bone is damaged. The ethmoid bone is like if you were to follow this nasal bone back into the skull, it's kind of in between your two eye sockets in the skull. If that is were to get damaged through some type of head trauma, then oftentimes you will lose the sense of smell. Damage to the temporal lobe can also cause a loss of the sense of smell. This is actually a common thing that we see in people who have Parkinson's disease and people who have dementia. Brain tumors and seizures can sometimes stimulate the primary olfactory cortex and produce olfactory hallucinations. So people start to smell typically unpleasant odors that aren't there, like burnt rubber or, just a really cringy odor that's not in their environment, but they're constantly getting that interpretation. Taste is our last one. Taste buds are taste receptors on the tongue. Um, Lingual papillae are the visible bumps on our tongue, and that's where taste buds are actually located. Primarily, our taste buds are mostly along the sides of our tongue. Without fluid, this is an interesting fact, then we aren't able to taste. So if you dried your tongue off and then patted some salt on your tongue, most likely you would not taste the salt very well. We have around 10,000 neurons that are 10,000 taste buds, I didn't specify, that are constantly being replaced. Food molecules are dissolved into liquids such as saliva and bind to receptors on taste buds. Um, Chemicals in foods can either release ions that enter the taste buds or they can bind to different proteins on the taste buds to give us that sense of taste. We respond to salty, sweet, sour, bitter, and umami. Umami is like that flavorfulness. I feel like that's just a new one we decided to create, but people use it now. So generate action potentials that are conveyed to the brain through your cranial nerves. Nerve 7 is that facial nerve. Cranial nerve 9 is hypopharyngeal. No, glossopharyngeal, sorry. Glossopharyngeal. And then the cranial nerve 10 is your vagus nerve. Perceived in small gustatory regions of the sensory cortex. Much of flavor in food is conveyed by its smell. So again, without the sense of smell, food often loses its flavor. And then gustatory just means taste. So they're in the small taste regions of the sensory cortex. All right, that is all for us today. So if you have any questions, you let me know, and we will see you in the next lecture.